Hello, my name is Jee-Hyub, and with my colleague Senay, we are presenting on key concepts in text mining and its usages. In our presentation, we have 3 sessions. In the first session, is about what is text mining, and we're going to also explain methods, available tools, gold standard corpus, etc. In section 2, we will introduce Europe PMC Central, where you can find right articles for you, and then we will explain if you want to build your own pipeline, how you can do it. The last session is where my colleague Senay will present about the CTTV project, where our Europe PMC Central text mining pipeline has been applied. So let's start with what is text mining. Every day we use Google, and sometimes Google helps us to find right information and also lead us to a right place where you can do further search. For example, when you want to buy some product, you start with Google, and it will lead to Amazon, where you can really buy some stuff. And in scientific articles, there's PubMed. But sometimes Google is not good enough for what you're searching for. For example, you have a list of articles in large scale, and when you don't have time to read everything. So can you say, could you give me a summary? This is what Google cannot do. And sometimes you want to find articles where genus cancer used in method sections. And another example is finding articles where data is used and mentioned, and especially with links to get those data. So these are questions where using Google cannot answer questions. And for example, this is one good example where text mining actually can help you. So let's go back to the example I mentioned, where if you want to find all those articles where genus cancer, not disease cancer, is mentioned in methods section on Europe PMC Central website, if you just to search for cancer, that will give you around 4 million articles. If you can make a query where you can specify, okay, so I want to find a cancer specifically mentioned in methods and organisms, which are organisms, then it will give you only 90 articles. Text mining really can help you to find those 90 real articles from 1 million articles. So there are already some text mining tools already available for you, but in order to find a right tool, you need to define your problem first, and having a bit of text mining knowledge will help you to define your problems. On next slide, we will explain what is text mining and what kind of problems in text mining, which help you to find relevant tools for your problems. So what is text mining? According to Marty Hurst, text mining is the discovery by computer of new previously unknown information by automatically extracting information from different written resources. More practically, this means text mining can help you to find right documents and extract information automatically, and if possible, you can combine those extracted information, which leads you to find new findings. The first part is information retrieval, which will help you to find right articles and extracting. The second part is information extraction, where you can extract information in a large scale. So we will focus these 2 parts in the next slides. First, information retrieval is to find relevant articles. You are all familiar with information retrieval, and in fact, you Google every day. Also in life science, every day, a large number of new documents are published and indexed, and PubMed is, for example, one of those popular search tools for biologists. Search algorithms. In information retrieval, there are several such algorithms, such as Boolean model, vector space model, PageRank, and which is a facet search, which is more like document categorization. And the reason I'm introducing these mentioning these algorithms is knowing search algorithms behind the site you are using is important, because that make you understand search result returned by those search engines. Information extraction is a text analysis method that transforms unstructured data to structured outputs. And first, you have to define your own templates, which basically tell you what to extract from articles. This is very similar to a table. Information extraction tasks consist of three tasks: named entity recognition, relation extraction, and event extraction, which is the most complex and hardest problem. And in this presentation, we're going to only focus on the first two, named entity recognition and relation extraction. So, what is named entity recognition? If you search for Francis Crick on Google, besides retrieved pages, Google algorithm recognizes Francis Crick as a name and provides more information on him. This is one good example of named entity recognition. In biomedical domain, gene, protein, disease, organism, and chemical names are important, and these names can be recognized by text mining algorithm. For example, heme oxygenase 1 was recognized and linked to UniProt for more information on this website. Relation extraction. Another task of information extraction is relation extraction, and its task is to discover semantic connections between entities. For example, given a protein BRCA2, finding all the proteins interacting is a relation extraction problem. So far, we introduced what is text mining and text mining tasks. In the next slide, we will introduce some background knowledge which can help you to find right tools and develop your own tools. In order to build your own tools, you should know what text mining methods are available. For that, my colleague Senay will continue to explain what are those methods.
In this part, I will talk about methods, tools and services, document and format types, gold standards, and performance evaluation used in information extraction tasks. There are 4 different methods used to develop text mining tools for extracting information. One of them is dictionary-based method. This method involves in creating a dictionary from terminological resources and using it for name entity recognition problems. It's a context-independent approach. By using this method, it is easy to link annotated entities in text to dictionaries or ontologies. Previous studies utilizing dictionaries reveal that dictionaries are generally complementary, so it would be ideal to merge them before using. Another method is machine learning. This method can be applied to various text mining tasks such as name entity recognition, relationship extraction, article classification, as well as developing natural language processing tools, for example, a post-tagger. It involves in training a learning algorithm on a gold standard dataset, and therefore it's a context-dependent approach. Similar to machine learning method, rule-based method can also be applied many problems such as name entity recognition, relationship extraction, and article classification. Rule-based systems vary from simple to complex depending on the complexity of the rules used to implement this system. For example, a system utilizing co-occurrences of entities for extracting associations would be the simplest approach, and utilizing regular expression, heuristic information, or natural language processing tools would make the system more complex. It is also possible to combine the three aforementioned methods and create a hybrid system. For example, a machine learning-based, in addition to a dictionary-based method, can be used in combination in name entity recognition tasks. There are various tools and services publicly or commercially available for either direct use or developing new text mining pipelines. One such tool is Jee-Hyub, which splits a given text into its tokens. Tokenizer is a natural language processing tool, and it can be used for the development of other natural language processing tools, such as sentence splitters, as well as name entity recognizers and classifiers. Another one is a sentence splitter called LinkPipe. Determining sentence boundaries is very important, especially for relationship extraction tasks. Banner is a name entity recognizer, and Europe PMC section tagger is a tool that can identify different sections of a given full-text article. Jee-Hyub is a text mining service that can be used to extract and visualize protein-protein interactions. There are also platforms that can be used for developing text mining pipelines. EMBL-EBI is one of them, and there are some packages that can be used to develop machine learning-based text mining tools such as Weka. There are many tools available for using and developing new text mining systems in case needed. However, one important question to answer is how to choose or develop the right tool. There are a couple of points that we need to consider before attempting to use or develop a tool. These are shown in the table. Independent of whether we attempt to use or develop a tool, firstly, we need to have a good understanding of the problem definition and ensure that its performance meets the requirements. If we intend to use an existing tool, we should check whether it is easy to use, operates on the document collection that we think contains the information that we look for, provides up-to-date results, and available for a long enough period for us. If we intend to develop a text mining tool, then first we should check the available tools to see if there is any that we can adapt it to our requirements easily. Otherwise, we should choose carefully a suitable implementation method among the ones that I mentioned earlier. Choosing an implementation method depends on the problem definition. For example, if you are a protein-protein interaction creator and would like your text mining tool to bring you all the possible protein-protein interactions for your further manual analysis, you could develop a recall-driven rule-based system operating on the co-occurrences of proteins at the sentence level. As I mentioned in the previous slide, document collection type and format play a role in the tool selection and development. There are various document types such as abstracts, full text, electronic health records, patents, and social media. Documents exist in different formats such as text, PDF, and XML. We should do a little bit investigation in order to understand where is the information that we look for is buried and in which format. Performance evaluation is an important issue for tool usage and development. There are three commonly used metrics for evaluating performance of a text mining tool. These are precision, recall, and F-score. We can explain how to estimate these values based on an example. Suppose there is a text mining tool which retrieves relative documents based on a specific user query from a pool of documents. Obviously, the document pool contains the actual relevant documents to the user query, as well as the relevant ones. The text mining tool retrieves both relevant and irrelevant documents. The Venn diagram shows the overlapping documents between the retrieved set by the tool and the relevant set within the pool. The relevant documents which are missed by the tool are called as false negatives. The relevant documents which are retrieved by the tool are called as false positives, and the relevant documents which are retrieved by the tool are called as true positives. False positives, false negatives, and true positives are used to estimate the system performance in terms of precision, recall, and F-score. Precision presents the exactness of the system and measures the percentage of samples that the system identifies as positive are actually positive. It is calculated by the equation showing in blue. Recall presents the completeness of the system and measures the percentage of positive samples which the system identifies as positive. It is calculated by the equation showing in red. And F-score is the harmonic mean between precision and recall. It is calculated by the equation showing in green. There are two ways to evaluate a text mining tool's performance. One is automatic, and the other one is manual. Automatic evaluation is applied if there is a suitable gold standard corpus exists. Gold standards are expect creator datasets, and it's very expensive to develop them. It requires certain amount of time to produce annotation guidelines and create the data. Therefore, there is no gold standards yet for many of the text mining tasks. Manual performance evaluation is applied in such cases which involves random selection of a sample result set and hand creation by domain experts. In order to design a good performing system, for a dictionary-based approach, we should use a high coverage, up-to-date resource. Also, the quality of the entries in the resource should be considered. This resource should be screened for positive, generic, or ambiguous entries, since such entries would cause to retrieve many false positive results. For a rule-based system, we should ensure that the generated rules are up-to-date and complete. For a machine learning-based system, we should carefully choose a suitable learning algorithm among the many available ones, such as support vector machines and neural networks. Feature selection and setting up training parameters are also play a role in the system's performance in machine learning-based systems. Another parameter affecting the performance is the gold standard dataset used to either train a machine learning-based system or test any system. Quality of the annotations in a given gold standard is important. If the gold standard contains many generic annotations, the trained system would retrieve many false positives. If the annotations in the gold standard are not up-to-date, the trained system on such a gold standard would have poor recall on the real text collection due to many missed samples. Similarly, if the gold standard presents a narrow topic, its size is small or contains some underrepresented sample classes, the system would achieve very poor performance in real settings. Let's close this session by providing some take-home messages. Firstly, each real-world problem has its own requirements. Therefore, good understanding of the problem requirements is the key for choosing or developing a text mining tool. Text mining is not trivial. It is challenging due to many issues. Firstly, text mining deals with natural language, which is complex and unstructured. this data is ambiguous. For example, name entities are ambiguous with common English words within and between species. In addition, the literature has a massive volume, making text mining as a big data problem. Therefore, text mining can help us only to alleviate our problems. Computers cannot mimic human perception, and text mining deals with unstructured data, which actually requires human perception.
This is session 2. In session 2, we will present Europe PMC Central and how you can build your own pipeline. Europe PMC Central is a search engine for biomedical literature. It contains over 30 million documents, of which 3 million full text and over 25 million PubMed abstracts. Among 3 million full text articles, 1 million is open access articles. On Europe PubMed Central, we try to reach articles using several ways, like we do citations, we identify named entities, we provide data links, and also we do section level searching. On Europe PubMed Central, there is only one search box for both Medline abstracts and PubMed Central full text articles. Search results can be ordered by relevance, date, or the number of citations of that particular paper. On the list of retrieved documents, you can see what are full text articles. You can also select only open access articles among full text articles. On Europe PubMed Central, we mine named entities, and you can see those results for each article. For example, if you visit an abstract, you will see those different colors on abstract. If you click those colors, you will see all those named entities. For example, disease names, gene protein names, and species. Another view of text mined result, particularly for named entity recognition, is a summary table. You can have all those named entities mentioned in full text articles in a tabular style, so that you can, instead of reading the whole article, you can quickly go through what genes, disease names are mentioned, what Go terms are mentioned, or what EFO terms are mentioned quickly. Thanks to text mining, you can do semantic search on Europe PubMed Central. This is the example I mentioned in introduction, where when you want to have all those articles where cancer is a genus and mentioned in section, you can write a Boolean query like this, and this will give you those 90 rare articles which is related to your own query. On Europe PubMed Central, we also mine accession numbers for data citation. For example, in this article, we identified PDB accession number, and if you click the highlight, it will give you a box where you can find more details on that particular accession number. For example, if you click PDB structure on this box, you will get the structure of this particular protein. On Europe PubMed Central, there is a mechanism called external links, by which user can provide relevant URLs to Europe PubMed Central. For example, if you deposited data in Dryad and you know relevant articles, you can provide a mapping table between those data, you will deposit it, and those articles related. That will make index built on Europe PubMed Central, and you can make search such as, can I get all those articles linked to a particular data set? For example, on Europe PubMed Central advanced search setting, there's a section for external links where you can choose a particular data repository, in this case, Dryad. Then you can make a query like, can I get all those articles where biodiversity mentioned in title, and also those articles linked to data in Dryad. This query will give you all those articles. Europe PubMed Central also provides RESTful web services. All those features I mentioned in the previous slide, you can also use RESTful web services in order to get the same features on Europe PubMed Central. This is the URI for RESTful web services on Europe PubMed Central. If you want to know more details, please visit this URL. Thanks to text mining, now you have relevant articles found. Once articles are found, you're going to start reading. While you're reading, often you will highlight and annotate those articles. Wouldn't it be nice if you can share those annotations and to discuss with your colleagues? There are annotation tools for this. Your annotation also can help with improving text mining algorithms, which will help with improving search results. Annotator is an open source JavaScript library for annotating web pages. On the top of Annotator, Hypothesis provides annotation platform. This is an example of annotating an article with Hypothesis on Europe PubMed Central. You can see the highlighted part, which is commented by a user, and this is shared, and you can see other users also commented on that particular part. As I said, using Hypothesis like annotation tool, you can annotate your article and you can share those annotations with your colleagues. In order to use Hypothesis, you just need to install a plugin for major browsers. You just go to website and you can download and you can install those plugins into your browser. Sometimes tools available for text mining is not good enough for your problems. In that case, you have to develop your own pipeline. In this part, we're going to explain how to build your own pipeline. On this slide, you can see our Europe PubMed Central text mining pipeline. This pipeline takes full text articles as input, and the pipeline transforms those articles by several modules. First, section tagger. Section tagger identify different parts of articles into a list of sections like introduction, method, result, discussion, tables, figures, etc. Once you identify sections, then sentence splitter will be applied, which identifies sentences in each section. Once we identify sentences, we will apply dictionary-based taggers, which is the most important part in pipeline. This is the key part for named entity recognition. After dictionary-based taggers applied, you will have automatically annotated full-text articles. Our Europe PMC central text mining pipeline has been also used for several use cases, mainly for mining named entities and accession numbers. The pipeline has been also adapted for CTTV project in order to extract relations. Dictionary-based taggers are a key building block in our text mining pipeline. We have chosen a dictionary-based approach for named entity recognition because this approach is simple, fast, readable, and interactive with domain experts like you biologists. What is dictionary? Dictionary is a just mapping table from terms to IDs, as you can see in this table. For example, the disease inflammatory bowel disease has been mapped to the particular EFO ID 0003767 in this case. We call it this as concept. The same concept can be represented in different terms. For example, here like IBD, which is abbreviation of the disease name. This will be also mapped to the same particular ID. In order to build dictionaries, there are many tool resources, ontologies, and your own spreadsheets. In order to build a dictionary-based tagger, here we will use Monk JFA, which is a Java class library for fast and flexible text filtering with regular expressions. You can download this program from the following URL. In order to build a dictionary, first you have to choose from your table what should be the terms and what should be the IDs. Once you choose the right columns for terms and IDs, then you need to create MWT file, which is just an XML file for mapping from terms to IDs. For this presentation, we will build EFO disease dictionary. First, you can go to online lookup service developed by EBI, where you can find the right ontology for you. In this case, experimental vector ontology. Once you choose your own ontology, the next step is you need to choose subtrees of that ontology. To build EFO disease ontology, we have chosen disease subtree and phenotype subtree. You also need to choose properties which you can use for selecting terms in your dictionary. One example of the table we generated by choosing those subtrees and properties. And in order to build a dictionary, we used only two columns, which are ID and term. In order to get this table, you can use the API provided by OLS. And if you need more details, please visit the URL mentioned here. Once you have a table and once you choose the relevant columns for your dictionary, now it's time to create an MWT file. This is just an XML file where you can specify those mappings from terms and IDs. In this file, there are two key elements. One is template and the other is T element. Template describes the output to be produced on a match, and T describes a dictionary terms you are using to build your dictionary. From this table, we need to write the following lines where we specify what the output gonna be look like. And this line means whenever you identify something that will be marked as disease. These are the list of terms from this table. For example, we put Alzheimer's disease with the corresponding EF4 ID. And we did the same for inflammatory bowel disease and IBD with different EF4 ID. Once you build your own dictionary, you can run your dictionary-based tagger against a sample file. Here I put one sentence in a file sample.txt. Then on Linux machine, you can run your tagger to that particular sample file. This is the Java command you can use once you download the Monk jar library and once you have your own dictionary. After running your tagger, you will get an output where gene, protein names, and disease names are identified and marked using XML element. If you want to know more details, this program has help page which you can refer to.
Session 3. In this part, I will provide you an industrial project as a sample text mining application. CTTV stands for Center for Therapeutic Target Validation, and it is a consortium of 3 founding members, including GSK, which is a pharma company, EMBL-EBI, and Wellcome Trust Sangha Institute. And recently, another company called Biogen has recently joined them. The aim of CTTV is to integrate evidences from various resources, such as gene expression and variation data, and also text mining data, and provide a publicly accessible platform for target validation. In our text mining project, we use Europe PMC as the literature repository and extract only the open access part of Europe PMC, because due to the licensing agreements, we can text mine only open access part and extract gene disease relations from articles and submit this data to the platform. Our project consisted of 3 phases. Phase 1 called pilot study. In the pilot study, we focused on a single disease, which was inflammatory bowel disease, and extracted gene disease co-occurrences at the sentence level. We filter out obvious false positive and estimated the performance. And in the second phase, we extended the study to 18 diseases identified as priority diseases by GSK. And we re-estimated performance to be sure that the system performance is stable. In phase 3, we further extended the study to all 8.5K diseases from EFO. We developed and used a disease name abbreviation filter, protein name abbreviation filter, to filter out false positives from our results. We also used a negative sentence filtering, and we introduced article scoring. There are 2 core dictionaries used in this project. One of them is UniProt for identifying genes in text, and another one is EFO, experimental factor ontology, for identifying diseases or phenotypes in text. However, we applied some refinements before using the dictionaries. We screened dictionary entities for possible false positive terms. For example, some of the entries are ambiguous with common English words like large, or other domain-specific terms like human stem cells. We screen out all the ambiguous terms, and we also screen out all the terms having length less than 2, because such terms would introduce false positives. We also generate new terms by replacing Greek letters with their symbols. For identifying genes, we use 2 dictionaries. First, whole UniProt, and the size of this dictionary is around 400K. And later, we use only human part of UniProt, and the size of this dictionary is just 100K. Our disease dictionary size is 26K. We applied filtering rules in 3 rounds. In the first round, we didn't apply any filtering. In the second round, we used only human part of UniProt because we realized that using whole UniProt would introduce us lots of false positive. And we also ensure that the long form of a given disease name exists in the text in case the abbreviation of the disease name appears in text. We also focus on only research type of articles, since other type of articles, like review articles, would introduce false positives. We also screen articles according to their sections. We filter out some sections, for example, acknowledgement, abbreviation, and methods, due to the fact that they would introduce false positives to the results. In the third round, we filtered out all the target disease associations that occur only once in an article body, but not in article title or abstract, because the co-occurrences in article title or abstract would be the most confident co-occurrences. This graph shows the results from our filtering. In the first graph, you see that we reduced the number of publications from 8,000 to around 3,500 articles. And in the second graph, you can see that we managed to reduce the number of targets from 3,000 to around 900. We estimated the system performance manually. For this purpose, 3 different IBD experts annotated randomly selected 45 records manually, and the precision of the system was estimated as 97%. In the second phase, we extended the study to 18 different GSK-focused priority diseases. Some of those diseases are Parkinson's disease, inflammatory bowel disease, diabetes, epilepsy, and cardiovascular diseases. We re-estimated the performance to be sure that the system is still stable, and the precision was estimated as 78%. In the third phase, we further extended the study to all 8,500 diseases from EFO. In this phase, we introduced disease name abbreviation filter. Some disease names, for example, ALS, means amyotrophic lateral sclerosis, is ambiguous with advanced life support in some of the articles. And you wouldn't like to annotate advanced life support as ALS. In order to identify potential abbreviations in text, we use regular expressions and check for the text sequences showing in red. We call this the former part, and we call this as potential abbreviation if it is tagged as a disease term by our dictionary, if it's in the uppercase form, and it has length less than 6. And we check whether the long form of the abbreviation from EFO exists in the document, or if the long form contains any of the keywords that were selected by our experts. And for examples, these keywords are disease, disorder, syndrome, defect. In this phase, we also filter out sentences mentioning on negative associations. Due to the complexity of natural language, it is difficult to develop such a filter. And the only clear evidence for us was the keyword not associated. If a sentence contains not associated, it's filtered out automatically. We also introduced protein name abbreviation filter in phase 3. Some of the protein names are ambiguous. For example, GCP, green cone photoreceptor pigment, is ambiguous with good clinical practice. Similar to the disease abbreviation filter, we use regular expressions and check for the text sequences showing in red. Again, we call the former part as potential long form, and the part appearing within parentheses as potential abbreviation, if it is tagged as a protein name by our tagger, if it is in the uppercase form, if it has character length less than 6. We check whether the potential abbreviation appears more than 3 times in document body, or the long form matches with any term in SwissProt or enzyme database, or long form ends with some phrases like is, eases, or it contains any of the keywords such as factor, receptor, gene, protein. Or at least 4 sentences contain one of the keywords like mutation, SNP, variation, gene, along with the protein name abbreviation candidate and a disease name. We evaluated the performance of our abbreviation filtering and negative sentence filtering steps, and we obtained an F score of 92.3%. We introduced article scoring in this phase because we wanted to score articles according to the confidences for given gene disease association. Since we have 2 types of documents, which are abstracts and full text, we implemented 2 different scoring algorithms. For abstracts, we assigned weights according to the sentence locations. For example, by our manual analysis, we figured out that the last sentence would be a result sentence. Therefore, we assigned result sentence the highest weight as 5. The second sentence or first sentence usually is an introductory sentence, so we assign it the lowest weight as 2. We also take into account the number of targets in the article. And since article abstracts lack in body, we added a boosting up factor to the score. And this boosting up factor is the median value of the article body scores from the open access set that we obtain. For assigning scores to full text articles, we use section information. For example, title would be the most confident part. Therefore, we assign the highest weight 10 to article. And we assign the lowest weight 1 to introduction, because introduction usually contains background information, and this wouldn't be a new and interesting finding for scientists. Since we introduce article scoring to the system, and we assign the lowest weight 1 to introduction, because introduction usually contains background information, and this wouldn't be a new and interesting finding for scientists. Since we introduce article scoring to the system, we needed to switch to another performance metric called mean average precision. MAP takes into account the relative order of the documents retrieved by the system and gives more weights to documents returned at higher ranks. We randomly selected 2 different associations and used them to estimate the MAP performance. We selected top 25 documents because we thought that the top 25 documents would be the most interesting documents for the user. We evaluated them manually and estimated the MAP as 81%. Now I would like to provide some statistics on the data. As I mentioned before, CTTV integrates evidences from different resources. For example, it integrates marketed drugs from Campbell, affected pathway information from Reactome, somatic mutations from cancer genes, and mouse models from Phenodyne. And interestingly, Europe PMC provides the largest amount of data. Currently, we have more than 3 million gene disease associations submitted to the platform. This table shows the top 3 diseases and top 3 targets extracted by using text mining methods. Neoplasm, cancer, and inflammation are the top 3 diseases in the platform, and TNF, IL-6, AGFR are the top 3 targets in the platform. Currently, we work on expanding EFO. For this purpose, we use experimental factor ontology, UMLS, which stands for Unified Medical Language Systems, and disease ontology to annotate literature. We extracted triples. Here the triple means gene, PMC ID, and disease term, and document the overlapping triples between those resources. You can see those overlapping triples in the Venn diagram. And interestingly, we have many more triples in UMLS and disease ontology. These triples are submitted to EFO team for the further expansion of EFO. In the future, we will continue with contributing to EFO expansion by using other ontologies like ORDO, which is a rare disease ontology, mammalian phenotype, and human phenotype ontology. We also would like to extend our study to identify mechanism type article type, and experimental type. Europe PMC CTTV component is available at https://www.targetvalidation.org. Please use your favorite gene or disease and give a try. If you have any feedback, we would appreciate to hear it.